yes, so this platform is not new because I think last conference also we all were there. So I had a golden chance to meet with Dr. Praveen Poddar. I have heard to Dr. Pushpam and Padma ma'am, and there were so many other eminent speakers are there, including Ramanlal sir as well. So again, this time we have again come up, and this seems to be a very perfect platform where everybody is experiencing, sharing their knowledge. My students have also been presenting, and it's a very good platform actually for the students. Like we as researchers, we as presenters, I think we had given a lot of opportunities, but the students are presenting, and I feel that that's a very nice thing. So the topic for my today presentation is a very different one. I am sure that most of you must not have heard about it. We know about cancer since a long time, right? Cancer being a notorious disorder, cancer being one of the very difficult disorder where each and every person or the patient is experiencing some unique phenotypes. The reasons are different, and thereby the clinicians, the doctors, they're facing so much of problem, like how to tackle this particular issue. So there was a new concept that has come up that that has just initiated reading past few months. I have not initiated any of my research work, so I'm not going to present any of the research work because I'm basically working on drug repurposing in oncology, and I think some of my students have been fantastically presenting my work. So I thought, let's not talk about drug repurposing for a change. Let me talk about something very different, something very unique. And since there are so many youngsters on this particular platform, there are so many professors, eminent professors, researchers are there. This might, give them a unique idea to initiate some different kind of research, so that even I'm planning to further go ahead with my career. So the topic, as given, the brief introduction. So I'm Dr. Jyotirmoi Aich. I'm working as an assistant professor in the School of Biotechnology and Bioinformatics. It's a DY Patil Deemed University, Navi Mumbai. I did my PhD from Institute of Genomics and Integrative Biology from Delhi. I did my postdoctorate in functional genomics and cancer under Dr. Amit Dutt in ACTREC. Post my postdoctorate, I have been associated with DY Patil University, so it's a fantastic chance to interact with the students, to work with the students. You learn so much from the students as well. So I feel feel very satisfied to be at this particular place. Plus, initiated, I have my own research group where there's a PhD students working, and we have this entire plethora of research areas. So the topic, coming down to our topic, it's a cancer cannibalism. I know the word cancer cannibalism, especially the cannibalism, I think it must have heard by a lot of people. We have so many movies also, we have some Hollywood movies based upon that as well. We have known about this, but how come it is related to cancer? In fact, the moment I have heard about it, it was something very unique to me as well, and that is it again have instigated that awareness and everything. That quest to learn something about more, and that is the reason I thought of sharing this information. So this is basically about all the information that has been known about cancer cannibalism in a very simple terms, so that we understand what exactly it is, and what we can summarize from this, and how research can be dependent on that particular part. So cancer cannibalism is one of the unique strategies by the cancer cells to survive chemotherapy. So cancer cells are very smart, they will do anything to survive this chemotherapy. They can, they have a lot of hypotheses, theories published, cancer stem cells, drug resistance, chemo resistance. There are so many, different strategies that these cancer cells are employing. And that is the reason, they employ some of this unique strategy that the researchers don't know, that the doctors don't know, that the clinicians don't know. And again, we have got this relapse. That's one of the very sad part of the cancer. Okay, moving on to the next one. So there's a very nice name I have given, in fact, this particular name. I have seen also a little modified. So cancer cells, they're feasting in the name of survival. Okay, so how come they are feasting in the name of survival? That they're surviving, but sometimes they are, doing a lot of parties and everything. How that is possible, and what is the mechanisms involved, and how it can be targeted for the efficient therapy of the cancer? That I would like to enlighten in my talk. So cannibalism, the word cannibalism is derived from cannibals, right, which is Spanish name for the carib people, formerly well known for practicing cannibalism. It's a practice of humans eating the flesh or the internal organs of the other human beings that we all know. Cellular cannibalism, now something related to cells. Okay, since I'm talking about the cannibalism, definitely that means that if I'm talking about a cancer cell, then somebody can imagine that, one cancer cell is eating up the other cancer cell, like the neighboring cell or something, right? That kind of a thing. Again, so coming down to the cellular cannibalism. Cellular cannibalism is defined as the ability of a cell to engulf another living cell, leading eventually to the death of the internalized cell. So something it does that it internalizes the other cell and eventually resulting in the death of the cells. So cellular cannibalism, it is defined as a large cell. Okay, I'm going to show you a lot of pictures in that, which is going to make the concept very clear that what exactly, like how this cannibalism is going to happen. Is defined as a large cell enclosing a slightly smaller one within its cytoplasm, and this is a morphological feature extensively seen in aggressive malignancy. So based upon this, so if a pathologist is identifying a cell being 2-3 cells are being inside one cell, they can come to a conclusion that this could be an aggressive malignancy. Aggressive malignancy is also going to talk about the metastasis pathway. Right now, we all know that the cancer, again, there's a benign, then there is a malignant, then there's a metastasis that is going to happen, and metastasis is the one which results in so much of devastating effects and all. So cellular cannibalism has been referred to as a cellular phagocytosis, right? So this term, the phagocytosis, I think everybody must have heard about it. So we know that, engulfing another cell, it would be a phagocytosis. So again, this also has been done, but again in a different scenario. So we have a cellular phagocytosis, we have a cell phagocytosis, cell-in-cell appearance, there is a cell-in-cell pattern, one cell delicately wrapped around the next one, that is going to happen. And the phagocytosis of the tumor cell by tumor cell again. So one tumor cell is going to phagocytize the other tumor cell, or a tumor cell embraced by the another tumor cell. So there have been a lot of research happening, although the research is not at that particular stage where people have actually identified that which all genes are responsible. Still, some genes are being deciphered, like they play an important role in cannibalism, but still kind of a ring was very much, distinctively very popular owing to their appearance. Now, cell cannibalism in a case of infiltrating duct carcinoma of the breast. Now, initially, people have identified like what are these different. Now, for example, whenever there's a biopsy, if any patient is complaining of any kind of a tumor or something, a biopsy is being taken, and then the H&E staining is being done, or there are so many other tests are also being done. So the pathologist identifies or see the slides very carefully that what exactly is the infiltration of the cells that has happened. So if they are going to observe that something very unique is there, there's a big cell which has encompassed 2-3 other cells, they can come to a conclusion, or they can do some more tests and everything, they can come to a conclusion that a cannibalism is happening. So if a cannibalism is happening, it can be confirmed, it can be said that this is at a particular stage, although those things are not at all clearly criteria is not made, or it has not been reached to the medical stage, because there's a lot of research still it is happening. So there's a in between, there's a big cell where you can see the small cells are there. Again, it's a case of, again, a cell cannibalism. In the case of cancer, let's move on to the cancer. The different terminologies like cell phagocytosis, a giant cell carcinoma of the lung has been observed. Cell in cell appearance, like there is an endometrial stromal sarcoma was there in which they've identified, and one of the scientists coined the term cell in cell appearance. There's a phagocytosis of a tumor cell by another tumor cell that has happened in the small cell carcinoma of the lung. Now, the cancer cells being, again, as we have discussed, one of the smartest cells, they resort to cannibalism. The cancer cells devouring their neighbors to survive the drug treatment is an abhorrent concept, okay? Yet, it holds hope for exploring new anti-cancer treatments, because we want those cells which are surviving this, any kind of a radiation therapy or a chemotherapy or a surgery. The basic target is that how to tackle all these cells, because these are the cells, if you have, for example, a surgery, there's an area where there was a tumor, a surgery has happened, there were some cells have been left out. These cells will multiply, and after 6 months, when the patient is again going to come for a CT scan or something, they will identify, okay, oh my God, at that particular position, again, there's one more, a tumor has developed. So there have been a lot of strategies, a lot of research across the world. Like, this is not I'm just talking about, there have been so much of research, going everywhere. We had so many talks also. Dr. Praveen Poddar has, immensely involved in all these cancers, research and everything since a long time. So again, throughout the world, people are identifying strategies, targeting, chemotherapy or any kind of therapeutics and all, which can prevent these cancer cells to proliferate. So cancer cell cannibalism is currently defined as a phenomenon in which an ensemble of a larger cell containing a smaller one. So that's important. We have got a larger cell in between. It is going to be accommodating a new cell, often in a big cytoplasmic vacuole. So there is a big cytoplasmic vacuole. I'm going to show you some of the stages, like how it is going to form. So it's detected in either culture tumor cells or in a tumor sample. So this has been detected. Cannibalism and the cancer. So cannibalism is complete. Now, whenever we talk about all these, hypotheses about death of the cell, okay. So the first mechanism that comes up is apoptosis, right? Then we come across necrosis, or we understand about phagocytosis. There are two more terms also involved, like entosis, that we're going to talk about, and there is a empirolysis is also there. So there are so many different mechanisms, everything leading to the cell death, but we have different mechanisms activated. So this cannibalism is completely different entirety than phagocytosis, entosis, and empirolysis. It is an important morphological feature to distinguish benign from malignant lesions. So there has been a lot of research which confirms that this can be used as an important morphological feature to distinguish the lesions. Cannibalism has been described in various cancers, such as we have bladder cancer, breast cancer, lung cancer, and this is all related with the aggressiveness of the malignancy. Now, is cannibalism being seen in the benign lesion? Yes, the answer is yes. The cannibalism in its true sense has been observed and described as an exclusive property of the malignant tumor cells, as we have talked about that aggressive malignant cells, they do show these kind of responses. The only phenomenon that may resemble tumor cannibalism in the normal human cells is autophagy, right? That occurs under a particular condition of starvation. So there was a very nice presentation by Priyanka before this presentation talking about the autophagy, the importance of the autophagy in the cancer. That's again, one of the things, like, it engulfs and it plays an important role in the cancer. Something, because we know that cancer is a multi-gene disorder. It's not specific, like we have this got one pathway that is leading to the cancer. There could be so many pathways, there could be so many crosstalks, there could be so many interlinking pathways. So we need to identify a particular therapy or a particular target that can be a kind of a common in between all these. I know the scientists have been working upon this, and I'm sure and believe that with the new technology, with the high throughput screening technologies, it will be possible to identify some kind of a permanent cure for the cancer. Let's hope for the best again. Now, there was one research article, like Osaki et al. They noticed that the benign cannibalism PARGD, this is a disorder, showed an immunological positive staining reaction to vimentin in all the cases. So this they wanted to confirm that whether it is from the benign or it is from the malignant. But there was no such staining in the malignant cannibalism in the transitional cell carcinomas, that is a TCC. So again, the link between the cannibalism the cancer, it is one of the important morphological feature to distinguish a benign from a malignant lesion, an emerging indicator of both the anaplastic grade and invasiveness. See, you do any kind of a research, okay? At the end, we want that a patient who is suffering from this terrible disease, some kind of a translational knowledge could be passed on, or any kind of a, so either you devise something which can be, could be a good indicator, or you tackle this drug chemo resistance. There have been so many other pathways, or you tackle with the cancer stem cells and everything that again Ankita has mentioned in her presentation with the help of antipsychotic drugs or something, the cancer stem cell property, or any kind of a chemotherapy like a therapeutic targets actually. So cannibalism has been related to the metastatic capabilities of the malignant cells. I have got one video that I wanted to show, but I'm not very sure that whether it is going to work or not, because I'm facing a little bit of internet issue. Let me try with this particular thing. I'm not sure. Actually, it was working, but then my... Yeah, I think it is working. So just have some patience, dear audience, because it's a very nice video was there. So I wanted to show all the the audience who are there, but I'm not very sure again. Yes, it has worked. So can somebody confirm, is it visible on my screen? The organizers? Hello? I'm not sure. Like, I think the voice is not there. I don't know. Like, okay, fine. So let me not just quickly, just a 42 seconds video. It's not a big one. So if I can show, you just need to observe the cells which are going to be there. They have done a time lapse imaging of a cannibalism with the cell fusion. I don't know why it is coming to be so small. So can you see there were two cells initially, and then there was a merging of the cells that has happened, right? And then there's a dilation of the engulfed cell that is going to happen. And finally, it has become the cannibalistic cell which has engulfed so there's a bird eye structure. I hope it is visible. I know it's very small, but I hope it is visible. Yeah, there's a bird eye structure. So that is, so initially there was two cells were there, and then there was a engulfing of the cells has happened. Finally, it has got a bird eye structure. That was the thing I wanted to show. Okay, moving on to my next slide. So why some cancer cells become a cannibalistic cells? The acidic microenvironment which is created in carcinogenesis due to a shift in the metabolic pathway encourages a selection of certain cell phenotypes that are able to survive in this caustic environment which is to the detriment of the other cell types. Okay, now these malignant cells are highly virulent and cannibalize that is the reason I just wanted to discuss that. What is the reason behind the survival of these cannibalistic cells actually? Now, in this case, whenever there is kind of an adverse condition within the microenvironment, such as hypoxia, there's a starvation, like a no nutrient supply, or acidity, some selected malignant cells have become highly virulent and they cannibalize or engulf their siblings, right? And again, they have such a report that they're also known to engulf some cells from the immune system as well. That is the thing that is going to happen. Now, coming on to the different types of cell cannibalism, it could be a homotypic of a cell engulfment, or there could be a heterotypic of a cell engulfment. Now, in this case, the homotypic, there's a epithelial cells that are white blood cells. Like epithelial cells are known to, engulf the white blood cells or the fibroblasts, as I have just discussed, is that they have been already information that they are known to engulf the immune cells as well, the neural cells. And in the case of heterotypic cell engulfment, we have this breast carcinoma or a gallbladder carcinoma, lung carcinoma. So there have been so many evidences in different cancer therapies where, there is a specific feature of cannibalism, which is being observed. Now, coming on to the morphological features of cellular cannibalism. So, cellular cannibalism in cytological or histological preparation, the crescent-shaped nucleus. So that is one of the typical morphological feature that to be identified. There is a crescent-shaped nucleus. This particular appearance is attributed to the fact that ingested cell is contained in a big vacuole that pushes the nucleus of cannibalistic cell to the periphery, right? This is going to happen. That is the reason there is a bird eye structure, right? So I think many students have done this embryonated hen sex, so where we identify, we have this, at the corner, the eye is being located. Now, that kind of an appearance again is being observed in a cannibalistic cell. So this particular appearance is attributed to the fact that ingested cell is contained in a big vacuole that pushes the nucleus of cannibalistic cell to the periphery. Now, quickly having a look on the schematic representation. So we have a cannibalistic cell, there is a free cell, there's a contact of the free cell with the cannibalistic cell, there's a gradual interrelation of the free cell into the cannibalistic that we have just shown in the video. Again, there's a complete interrelation of a free cell into the cannibalistic cell, leading to a crescent shaped of nucleus of the later cells, and there is ultimately the interlaced cells is going to free cell is going to die off. It's a similar structure again taken from a different paper, which is going to talk about in a different manner. So we have a free cell, we have a cannibalistic cell, there is a engulfment, there's a internalization, and then at the end there's a crescent shaped of nucleus. This is a nice picture that I got it again from one of the paper where they were trying to show the different stages actually as it is. This is the actual part. Like this has been done in a breast cancer. Of the breast duct has been taken. There has been a cell cannibalism has been there. So initially there would be a one cell. Then how the fusion has taken place. In this. Then there is a bird eye structure that the engulfment and everything is going to happen. The engulfment and everything is going to happen. And then again, the another cell is going to engulf that particular thing, and then this got further divide. So a cannibalistic cell again, it can go for a further division also. That also has been observed in some of the cell types. So pathogenesis, we have, as we just discussed, that there could be hypoxia, there could be a lot of stress thing, that could be also because of an acidic environment, because of a lack of nutrition supply, because, there is a competition. There's a competition happening, when we talk about the survival of the human, when the Charles Darwin they were like, the one who is going to be, the most strong is going to survive. The same thing is going to happen inside our human body with the cancer cells as well, that if there is going to be a competition for the nutrition, then these are kind of mechanisms people have observed. So lack of nutrition in acidic environment, activation of specific lytic enzymes, and there are some proteins also being revealed out, like we have caveolin, we have a cathepsin B and azurin. There's a weakening of intercellular membrane junctions, which is going to happen of your, both the cells is going to happen. Release of cells into the intercellular matrix. There's an interaction of the cannibalistic host cells and the weakened cell, and engulfment and entrapment of the weakened cell by the cannibalistic cells. Apart from that, there is one more mechanism that has been observed called as a complex cannibalism. Okay, now what could be a complex cannibalism? Cannibalism already it is complicated, but to add to the complexity, there's one more cell which is engulfing this cannibalistic cell, which would be called as a complex cannibalism. So a malignant cell which has engulfed another malignant cell might in turn get engulfed by a third malignant cell. So this could be one of the example. I think there's a nice picture again in the next slide. The process is referred to as a complex cannibalism. So if I'm going to show you in this case is that there's a one malignant cell which is engulfing another malignant cell. So if you can see that now and this complex is engulfed by another cell in the B and C. In the B and C figure you can see. So again in this case there's a small cell which is engulfed. Finally this cell is being engulfing the other cell, and thereby finally in one cell there would be two cells, right? So there's a third cell that is going to be here, and in the last one that is in the D figure you have one malignant cell which is going to have two cells, which is very clear actually. Again, this H&E staining are the clear true representative of whatever has been observed by the pathologist. Schematic representation for the malignant tumor cell cannibalism and a complex cannibalism. So we have this malignant tumor cells are there, and there is a WBCs. Remember, apart from engulfing the cancer cells, it is also known to engulf the WBCs as well. So in this particular case, there is a WBC engulfment that has taken place. So we have got one cell, tumor cell, which has taken up this WBC cell. After that, the malignant cell engulfment by another malignant. So this thing has been taken up by another cell, right? So engulf it, malignant cell engulfing another malignant cell. So again, this is all a little complicated phenomenon. Now, the best part is that, this can be clearly observed, right? So for example, when I see a H&E staining of a normal tissue where there is no infiltration at all, when I see a cancer cells where there would be a lot of infiltration because inflammation has happened, there would be recruitment of your W size, eosinophils, basophils, and everything is going to happen as the students must have read about in their immune system. And again, after that, if I see a cannibalism image where the cannibalism cells are there, now since it is very different. Now there's a crescent shape of nucleus that is going to form apart from a single cell, a typical round circular nucleus. So a pathologist or a clinician can really identify this to be a very precisely clear, which could be crystal clear. This was one of the report by Townsend-Murray adopted. Now, basically, what you have to do is that the researchers or the clinicians, they need to have some very good imaging techniques actually, because with a normal microscope and everything, it is possible that you might, miss out all these important things that are happening. So Townsend-Murray adopted elegant cell labeling methods using real-time microscopy to observe cellular ongoing by the drug-treated cells. So this was an in vitro culture. They discovered that in response to the drug treatment, cells that become cannibals were able to outlive their unindulged neighbor. So that is important. So they have taken the cells, they have given some treatment, and then they wanted to understand that what exactly is happening. So there's a very nice paper that has published cannibalism in the breast cancer, the dangers of overeating. So this has happened. This was again taken from a nice paper, which it was actually beautifully being seen in the breast cancer cells. So feasting in the name of survival. So these are the some of the cannibalistic tactics that are employed by the breast cancer cells in response to a therapy. So basically, what happens, whenever a breast cancer cells or any kind of a cancer cell is being treated with a drug, so it is going to induce a kind of a senescence, senescence, or there would be some other mechanism of the cells that would be activated. So these are the breast cancer cells, and the chemotherapy is going to induce some senescence to the neighboring cells as well. So this is going to result in the activation of senescence and phagocytosis programs, right? Now, following this, this could lead to an engulfment of the neighbors, right? Once the engulfment has happened, it can result in the lysosomal degradation, right? After that, depending upon what is the situation, it can go for an extended survival and persistence. It can go for a dormancy as well. It can go for a relapse as well. So depending upon, what kind of a phase that cell is experiencing. And then if a drug re-challenge is going to happen, how the cells are going to respond to that particular treatment, it can decide about what kind of a mechanism is there. Talking to the next one is that, we are going to talk a little bit about like a cellular cannibalism. So whenever, this cannibalism word is there, the first term, again, definitely we have autophagy, we have apoptosis, we have phagocytosis, we have entosis, we have empirolysis and everything. So I wanted to talk about, because we know about autophagy, apoptosis, I guess majority of the people must be knowing about it. These two terms are very, very a new novel one, because these two terms were new also to me when I was, actually reading about the cellular cannibalism or cancer cannibalism. So there's the entosis and there is the empirolysis. All these are closely related phenomena because all these are doing the same thing actually. But since they are very different in their morphology and everything, so it's easy for the people who are doing a research to catch them to catch a hold of them, right? So all these are closely related phenomenon with a very fine line of difference. Its corrected identification has a significant diagnostic and a prognostic value. And there is the take-home message, from this particular part is that if you correctly identify that, this can serve as a good indicator. If you identify the genes or any kind of a microRNA or any kind of a regulation mechanism which is affecting these proteins, which are important in cannibalism, you can at least stop these cells to survive from chemotherapy. That also can be done. So a little information about entosis and empirolysis, right? Now, entosis, it's a very, a new term actually. Entosis, basically, it's a cell eats cell world. Okay, so a cell eating another cell. So that kind of a mechanism that is happening. So entosis, initially it was identified by Michael Oberholzer. Now it was reported in 2007. So most of the animal cells require an attachment to our extracellular matrix for their survival. Within the mammary gland, death of the matrix-deprived cells underlies lumen formation during development and is thought to act as a defense against the formation of breast tumors. Okay, because there have been a lot of barriers and everything, and there is a layers of cells that have been deposited in the breast cancer, in the mammary gland. So probably it could be served as a kind of a defense. Cells detached from their extracellular matrix, I guess there's a sentence missing. Okay, cells detached from their extracellular matrix can undergo a process of entosis. The cell-in-cell phenomenon was seen centuries ago, but Overwalter et al. In the 2007 reported that detached epithelial cells can also take an alternative path to death, right? Which is driven by a cell-in-cell invasion, which was named as entosis. So entosis, basically in the Greek, if I'm going to bifurcate the word, it is entos, that is meaning inside or within, right? That means that, the moment, a cell, the fate has been defined that it is going to go for the death of the cell. Either it can go, the cells can die off. Either it can release some of the nutrients. It can go to another. It can a dying cell can again instigate another cell to die off, or it is going to engulf. So there are so many different complicated pathways that plays actually. So entosis, how it is going to be very different from the cannibalism. Now, entosis, it is a homogeneous cell invasion, like a similar kind of a cells are going to happen. But in this case, we have seen like a cell taking up the immune cell, or a breast cancer, any kind of a cancer cells taking up another kind of a cancer cell, that also is possible. So while invasion can be either homogeneous, or it can be heterogeneous. In entosis, the live epithelial cells or the tumor cells detach from the extracellular matrix and then invade their neighbor cells. Entosis rely on conjugation or adherence functions and need a row and row activities for internalization, suggesting that entosis is an active process and it requires actin polymerization. So quickly showing you the pathway, actually, the schematic representation for the phenomenon of entosis. Now, in this case, again, the proteins which are present on the surface of the cells is going to play an important role. It is going to play an important role, actually, that particular kind of mechanism that is going to happen. Yeah. So stimulation of entosis by oncogene KRAS. Okay, now these KRAS, the tumor suppressors, are going to be important. So these are the different classes of tumor suppressors are going to be there, which plays an important role. Now, in this case, now the cells are there, which has got some active receptors like E-cadherin or the P-cadherins are going to be there. Now, these are the cell attachment proteins. Okay. Now, cell-to-cell attachment driven by the cadherins are going to happen. Initiation of process engulfment. So initially the cell would be there. If the KRAS is activated, then this E-cadherin or the P-cadherins are going to get activated. Once it is going to get activated, there's going to be a cell-to-cell attachment driven by the cadherins, because once the cadherins are activated, they would be able to go and bind there and make the cells fuse together. Initiation of process of engulfment. Now, this is going to help in the engulfment of the cells. And at the end, you can see that engulfment for the cells in the vacuolated space. Okay, that would be there. One more mechanism that is again going to play an important role is called as a empirolysis. Okay, now empirolysis is a condition where the hematopoietic cells in a living and intact state are seen in the cytoplasm of the host cell without any damage. So that's again a very little different mechanism. Again, I have some schematic pictures with the H&E staining, which is going to be make you things little clear. Now, so coming out of this is a condition where the hematopoietic cells in a living and intact state are seen in the cytoplasm of the host cell. Now, the cells taken in frequently are neutrophils, we have lymphocytes, we have plasma cells. The host cells may be a megakaryocyte, monocyte, endothelial cell, fibroblast, and a malignant cell. In this phenomenon, with both the cells being viable, there is not any physiological and morphological sequences to either of them, right? That is the phenomenon that is going to happen. Now, this is a very peculiar picture of empirolysis. Like I have taken this H&E staining. So there's a disease called as a Rosai-Dorfman disease. There's a hallmark of this disease is called as a empirolysis. Now, in that what happens? The lymphocytes and the other inflammatory cells reside Okay, so you have lymphocytes, you have other inflammatory cells, which would be eosinophils, basophils. The different cells are there. They reside undistributed within the cytoplasm of the histiocyte. So they just sit inside the one of the cell. So if that kind of that kind of a scene is there, again, it could prove to be one kind of a disease. So impurity space then, and it has taken up every cell inside. Like the plasma cell is also there, the lymphocyte is also there, the macrophages is also there. So again, now this thing is again very different from a cannibalism, right? Again, if a pathologist is going to see, usko lagega ki haan bhai, if this cell is also engulfing, the endosis is going to happen. But you have to be very sharp in identifying what is the major difference among these three mechanisms. So Empirolysis of a red blood cells in a vacuolated space by megakaryocytes is again a different figure where the RBCs and your megakaryocytes are there. Just some cannibalism and other forms of cell engulfment are there. So you have, a kind of a comparison, because we have almost come to the end of I have come to the end of my talk. So talking about cannibalism, there is an empirolysis, there is a phagocytosis, there is an autophagy, there is a benign multinucleated cells, and there is an entosis. So we have a cell in the cannibalism, the cell within cell causing the death of the cell. Empirolysis, the cells are not destroyed. Everything is being happily staying together in one cell. Phagocytosis, pseudopores, surface ruffles are going to see. Autophagy, we have membrane autophagic vacuoles named phagosomes. Benign multinucleated cells, we have results of the chemotherapy or the radiotherapy is there. And entosis, it is a cell-in-cell invasion. So it's a very nice classification that we have done. So there's one, I think, presenter in our conference, Kartikeya is there. So Kartikeya and we are working on this, cannibalism. We are thinking of some of the important parameters can be important. So I think working with him again, it's a team actually. It's a big team that we have 2 faculties and so many students are working. So again, I think, within probably by the next year, I would be having some data where I would be able to share with all you people talking about the cellular, cancer cannibalism. Coming on to the end of my talk, so cannibalism is a completely different entity than phagocytosis, entosis, or empirolysis. The cannibalistic cells, they have the ability to cannibalize their sibling as well as the cells from the immune system, right? So interestingly, the metastatic tumor cells are also able to engulf and digest the living cells, including the autologous lymphocyte that should kill them, right? So that's the CD8 cytotoxic lymphocytes. Now this, at the end, we need to understand that what could be the opportunity, right? So this can represent a formidable opportunity for metastatic cells to survive in adverse conditions, such as those encountered in their journey towards the target organ to establish a colony. Again, altogether, these findings seem to suggest a pathogenic role for the cannibalist. So we have been talking about that, what is the mechanism, kaise ho sakta hai, why it is happening, and how it can be used. So again, this all are basically a pathogenic role. And the point at this surprising cellular aggressiveness as an innovative pharmacological target. So people can think of we can target any of the genes, protein, as I have discussed in my talk, in the clinical management of the metastatic disease. The engulfment process described is energy consuming. Definitely, it requires a lot of energy, but is predicted to return value to make the expenditure worthwhile. So definitely, as an energy demanding activity, this suggests that innovative drug targeting of this practice may offer new avenues for a cancer research. So this is entirely a new concept that has come out. I am sure that I would have been able to contribute to some of your new knowledge that is going to trickle your brain to think something about the cellular cannibalism as it has done to me. So with this, I would like to end my talk. I would like to give acknowledgement, not for this talk actually, but all for my research, but definitely to my